The Cincinnati Museum Center does have a DNA lab. That's one of the first questions I often get from visitors, being very surprised that we have this facility. So the photo here is what the lab looks like now, but I'm going to talk a little bit about its previous version. So the Museum Center's DNA lab was actually established in 2007. So we've had a DNA lab for over a decade. And it was primarily funded through National Science Foundation grants for a DNA sequencer and also donations from museum supporters for smaller equipment to outfit the lab. And when the lab was opened, it was housed in the Geyer Collections and Research Facility down the street from Union Terminal. So it was behind the scenes in our building that houses all of our collections. So the public really wasn't aware that we had this lab facility, and it certainly wasn't something most visitors would see. So some of the things that we do in the lab, we do all animal genetic studies. We don't do anything that's clinical or human related. However, DNA is DNA. So we use all of the same laboratory techniques and equipment that you would see in a forensics lab or in a clinical biology lab. And I'll talk a little bit more about that later in the presentation. We also do a lot of phylogenetic work. So looking at how species are related to one another. So if you've ever seen any of these basically family trees or tree of life that links different species together through time, that's basically phylogenetics. You're looking at the evolutionary history of species or different organisms to one another. My area of expertise is actually population genetics. So I look at things like genetic diversity in populations, migration, so how animals are moving from one population to another, looking at things like inbreeding, if genetic diversity is declining, how are those impacting the reproductive success of individuals? We look at population fragmentation. So again, how are animals moving or not moving between population patches? That's particularly important these days because of habitat fragmentation, where you might have wooded areas that are separated from each other by agricultural zones or cities or urbanized landscapes. So that has a lot of implications for how animals are able to move between fragments of habitat that are suitable for them. We also do some things with parentage. So looking at what animals are related to offspring. So we're doing a couple of studies looking at extra pair paternity in birds. So how often do females breed with males other than the ones that they're nesting with and that sort of thing. We also do DNA barcoding, which kind of works almost like the UPCs on your groceries. You can sequence a particular DNA fragment from the genome and compare it to existing databases of genetic information and try to identify the species that you have. And I'll talk a little bit about that in a few minutes as well. The photo on the right is some DNA sequence data coming off of the sequencer. So that is real-time data coming off of the machine as it runs. To give you an idea of the different species that we work with in the lab, we have some photos here of some of our current research subjects. Up here on the right is a crayfish. Emily Imhoff, my collections manager in zoology, studies crayfish, and she's particularly interested in the parasites that infect them. So she's actually using DNA barcoding to identify various parasitic infections that crayfish can get. This little guy is a white-throated sparrow. We're doing some genetic sexing projects on white-throated sparrows as well as blue jays in conjunction with a professor at Miami University. This yellow and black bird here is a hooded warbler. We're doing parentage analysis on that project. So the professor, there's a professor at Ohio University studying this species, and she wants to know how often are males at a nest actually raising young that are not biologically theirs. So we're really interested in seeing how often males are caring for other males' offspring. The snake in the bottom corner here is a short-headed garter snake. We're doing some work for the state of Ohio to examine whether or not we have breeding populations of short-headed garter snakes in Ohio. Currently, they're found right along the Pennsylvania border. So we're trying to establish if there's actual breeding populations in our state or if they're just migrating across the state line. And that has implications for whether or not it's a listed species. So if it needs to be protected as a threatened or vulnerable species in Ohio. This little rodent down here is an Allegheny wood rat. We've been doing some monitoring of our population on the edge of Appalachia Preserve in Adams County, looking at genetic diversity through time and how we should most effectively manage those populations. And then the insect here in the top corner is the spotted lanternfly, which is a new invasive species in the US that's heading this way. And I'm involved in a project with the USDA and Penn State developing genetic markers to be able to track how that invasion is progressing across the US. So we have a big variety of research questions, a big variety of organisms that we're using. But as I said, DNA is DNA. Once it's out of the organism, it's all treated the same way. So we can use the same methods to answer lots of different research questions about the ecology of different species. So the original lab was founded, like I said, at the Geyer Center. And it's not shown in this photo, but this is the old lab. And it was basically half of the room where we do our zoology prep activities. So we actually shared this space with the area of the building where we actually skin mammals and birds for inclusion into the research collections. That's not an ideal setup because there is the possibility of contaminating genetic samples that you're working with feathers and fur and blood and other body fluids from animals that you're dissecting. But because we were doing mostly work with tissue samples that are very, very concentrated in their DNA, the risk for cross-contamination was pretty minimal. The work surfaces weren't ideal. You can probably see from these pictures, we have lots of wooden tables, lots of folding tables with laminate tops. Not great for doing work with chemicals and DNA samples and whatnot, not very easy to clean. So that's why everything was covered in bench paper. And we also had pretty limited storage. So we had open wooden shelving for glassware and chemicals and all kinds of things. So not great storage space, but it was a functional lab. We made it work. The previous curator, Dr. Herman Mays, who founded the lab, had a very successful research program out of this little space. He made the best of it. So we had everything we needed to do the genetic work, but it was definitely a tight space with pretty limited resources. So here's a couple more photos of the lab. The lab was basically a single user lab. It was really difficult to get a group of people in the lab doing something. So you can see here, there's a group of students from a local university trying to learn some genetic techniques. So we didn't really have any redundancy in equipment, which is problematic sometimes when you're trying to teach students or let people do things simultaneously in the lab. Also, we had a lot of outdated and broken equipment. So this picture on the left is our old DNA sequencer. And when I took over the lab in 2016, this sequencer was approaching the end of its life. The model was being discontinued by the manufacturer, so we were going to be unable to get replacement parts for servicing on the machine. So that was a concern when I started, what we were going to do with our DNA sequencer once it became obsolete. And then, like I said, there were a few pieces of equipment that had been sort of MacGyvered to still function, even though they weren't working optimally. So when I started, I tried to replace a few of the very small pieces of equipment in the lab so that we didn't have that issue. So in 2016, we began the Union Terminal renovations, and the decision was made to move the DNA lab from the Geyer Center to Union Terminal, along with So to basically give us an upgrade in the space and the equipment, and put it on public view. So over the next couple of years, we got a crash course in how to read architectural designs or schematics and whatnot. We were trying to figure out how we were going to lay out the equipment in the lab, try to decide what our power needs would be, our plumbing needs, our ventilation needs. So trying to visualize what the space would look like, where things would go, and try to do our best to design the lab space the way that it would function best. So there's just a series of photos here I want to go through. This is basically the beginning stages of the lab, basically concrete floor with drywall. And over time, the ductwork and plumbing were added. The space started coming together. The walls were finished. All the electrical outlets and things were being installed. Our cabinetry was put in with our countertop that would have our sink installed. You can see a few pipes and things sticking out of the wall for our emergency shower and our plumbing and things. And then we had our fume hood installed. So we do work with a lot of chemicals that need to be properly ventilated out of the space. So we had our fume hood installed. We had an emergency shower and eye wash installed as well. And eventually the glass walls were put in. And we made a trip to IKEA to get some metal shelving for our plastic consumables. We acquired a glass-fronted metal cabinet for glassware and chemicals. We also had some office furniture delivered. So in the back of the lab is actually a data analysis area where you can sit with computers and work on analyzing the data that's produced in the lab. We had our sink installed with our paper towel and soap dispensers. Here is one of our TV monitors that was installed. So we are able to stream content related to the lab on these televisions. So right now we have basically a PowerPoint that streams to give people information about the history of the lab, the staff members, and some of the current projects we have going on. So as we were packing up the Geyer supplies to bring over to Union Terminal, we decided that we needed to have lab mascots. So this is Gene and Helix, our lab rat mascots. Helix is after the double-stranded helix of DNA structure, and then Gene, of course, is the genes in the genome. So we packed them up with all of our Geyer supplies that were moving to Union Terminal and gave them a look at their new home. They seemed pretty impressed. So our workbenches actually were delayed several months. So unfortunately, we sat with an empty room for quite a while. But finally, the day came when our lab benches were installed. You can see the guys here doing the installation. All of the lab benches are on wheels that lock. So we are able to move them around and change the configuration of the lab if we need to. They're also equipped with electrical outlets, so we can plug in small pieces of equipment throughout the lab, not just against the walls. And we also have these little portable storage units that we can change the configuration of as well, so we can move them where we need them. So this is the view that the public would get as they approach the lab right after our bench tops were installed. So we had finally our tables all set up. And it did not take us long at all to unpack all of the equipment that had been delivered over the past several months, get everything up and running. So setting up the space didn't take that long, but the really time-consuming part was after all of this equipment was unpacked, everything had to be calibrated, everything had to be tested to make sure everything was functioning okay, make sure we didn't have anything that was broken or needed to be returned or refunded to us. So that took a couple of weeks to get everything situated. And some of our larger pieces of equipment had to be professionally installed by technicians. So this is how the lab would look today if you went up to the railing here to peer in. One side of the lab is actually elevated on the outside, so the visitors actually look down a little bit into the space. But if you go around to the other side of the lab that is also glassed in, you're level with the researchers inside. So you can actually view the lab from two different sides. And we have Gene and Helix here in front of our welcome board, greeting visitors as they come by. So this is actually the second research lab to be put into Union Terminal. The other lab pictured here is the Paleontology Lab. So the Paleontology Lab did have a workspace prior to the renovation, but their workspace was moved and updated as well. So these two labs are actually right around the corner from each other at Union Terminal now. So you can see paleontologists as well as the zoology staff at work. So a few of our new toys that we got. So we got a major upgrade with moving the DNA lab to Union Terminal. We got our updated DNA sequencer. So the sequencer that was going to be obsolete was replaced with a newer version. So that should keep us for quite a while. We also got a next-gen sequencer. So the difference between these two machines, the traditional sequencer basically sequences one long piece of DNA in each of the reactions that it runs. So you can sequence up to about 1,000 base pairs or 1,000 A's, G's, C's, and T's that make up the genetic code, as well as doing some other types of markers that we use in genetic testing. But the next-gen sequencers are able to sequence millions and millions and millions of pieces of DNA simultaneously. So our particular machine, this little iSeq 100, will sequence about 4 million fragments of DNA at the same time. However, those fragments are very, very short. They're only about 150 base pairs. So what happens is you have to take those very small fragments of DNA sequence and basically reassemble them into larger reads. It could be chromosomes, it could be a whole genome, it could be lots of different genes that you're targeting. But basically you have to take all those little fragments and reassemble them like a puzzle. So it's really useful for certain types of studies. We've used the next-gen sequencer so far in the lab to look for new genetic markers on the spotted lanternfly project. So we've done a few runs on the machine to generate new markers that we can examine for those studies. So that's really exciting. So I'm really hoping I can get more into next-gen sequencing methods and projects than I have in the past. We also have a real-time PCR machine, also called a qPCR machine, which would be quantitative PCR. This is typically used for things like disease screening. It's used in both zoology context when we screen for things like chytrid fungus in amphibians or snake fungal disease in reptiles. But real-time PCR is actually the technology they're using to process the COVID-19 testing samples. So you hear all about these swabs that are taken from potential cases and they're being tested in a lab. And real-time PCR is actually the method that they're using to process a lot of these samples. So it's really neat to have that link in our lab, the same technology that they're using in the pandemic. And of course, you probably saw from the photos before, we have our space is definitely multi-user now. We have a lot of redundancy in equipment. So a lot of things can go on simultaneously and people can work at the same time without competing for equipment or getting in each other's way. So some of the goals for the lab, we want to show the public that the museum is not just a place to display cool objects and storehouses of very cool specimens and things. It's actually an active research institution. So we want to show the public that we are contributing to scientific knowledge. We're trying to stay involved in the research community, and that we're not just showing stuff at the museum, we're actively increasing scientific knowledge. Another goal is to educate the public on genetics and the projects that we're doing, all kinds of things related to what's happening in the genetics field. We're kind of inundated in the news with all kinds of talk about genetics and personalized health and that sort of thing. And eventually we'd like to get more information involved with those types of topics to educate the public and help them better digest the news that they're hearing from other sources. And of course, we want to mentor students and young professionals. So people who are looking to get into these types of fields or work in these types of environments. So mentoring students is going to be really important down the road to get some experience through internships or volunteering to learn their way around a lab. A few challenges. When we first opened the lab, one of the things that I was hearing a lot as I was watching visitors come by the lab is they would always say, is this for real? Is this real? Is this just a display? You know, what is this? So I took our whiteboard in the lab and did this little message on it here, and it says, is this for real? Yes, in big block letters, this is absolutely for real. So I have written on here that our museum scientists use this lab to study all these different things. So I don't know if this is just a general misunderstanding when people visit museums or if this is a carryover from our old exhibits. Prior to the renovation, we did have a few sort of imitation labs where you could look and see what an ornithology lab would look like, or an archaeology lab would look like, or a collection space would look like. So I don't know if that's a carryover from our previous exhibits, or if this is just something that people aren't used to seeing at a museum, to actually see a real functioning research space on the exhibit floor. Communication with visitors is a big problem. We don't have anything like an intercom where visitors can directly interact with us or ask us questions or let us narrate what we're doing in the lab. And visitors are not allowed access to the lab, so they don't come in when we're working. So communicating what's happening in the lab to the visitors outside is a continuing challenge, and we're going to have to work on that. We do have our TV monitors that, like I said, stream some information about the lab, but hopefully down the road we will get some exhibits that are outside the lab that will give visitors a little bit more information about genetics and the things that we do in the lab. Of course, lab staffing and funding are issues. So right now, my collections manager, Emily Imhoff, and I are the only ones on staff who do genetic research. So it's difficult for the two of us to have things going on in the lab all the time. And the other thing is funding. Genetic research, although it has really decreased in cost over the last decade or so, it's still very expensive. So a lot of things that we would like to do in the lab are sort of cost prohibitive unless you have external grant funding or someone who comes to you with a project and they've got some money they can contribute for reagents or for staff time. So we're hoping to really work on attracting more grants and more funded projects in the future. So speaking of funding, I just want to recognize our initial donors to the DNA lab, Irvin Melinda Simon. They gave us a sizable donation for us to outfit the new lab. So I just want to recognize them. They got all my new fancy toys for me, so I appreciate that. And with that, I just want to say thank you everybody for joining me today. This is actually an image from our opening party. When we reopened after the renovations, we had a projection mapping done on the front of the building, and there was a DNA strand rotating across the dome of the rotunda, which was very cool. So I want to thank you all for joining me. If you want to see more content from the museum, more videos and interactive activities and things, you can check out our website at cincymuseum.org. Also, as you know, during the pandemic, we're closed, so our revenue streams are greatly reduced. So if you'd like to help us out, we encourage you to renew your membership or join us for the first time. Also, we have a COVID-19 fund for donations that you might want to make, and those are all on our website. So I encourage you to check out the website, our social media accounts. There's all kinds of things going on every day. that you can tune in to keep in touch with us. So thanks again for joining me. And if you have any questions, I would be happy to answer those.
I have a question.
Sure.
What kind of precautions do you guys take to minimize the risk of contamination or cross-contamination?
So we use, of course, sterilizing surfaces and equipment. So we usually bleach things down in between experiments. We can use bench paper that can be disposed of so we don't contaminate our surfaces. We also have a UV sterilization chamber in the lab, so we can actually put our pipettes and small items into a UV chamber that will basically damage the DNA that's left on anything. Typically, it's not too big of a problem because, like I said earlier, we're using basically tissue samples that have very, very highly concentrated DNA. So if there is any cross-contamination, it's going to be swamped out by the samples that you're working with. But also, a lot of the projects that we do, the genetic markers that we're looking at are specific to the species. So if we would get wood rat DNA into a bird sample, we're looking at bird specific loci, so that wouldn't be a problem necessarily. But we do, you know, like I showed, we do all kinds of different projects on all kinds of different organisms. So we do have to be careful to not cross-contaminate our samples.
And why is it not possible to work with ancient DNA then in these types of situations where we could, it sounds like a lot of the precautions are basic everyday disposable items or over-the-counter type items like bleach.
They are, but the problem with ancient DNA and doing samples that are very, so basically once a cell dies, the DNA inside of it starts to degrade or break down. So what you end up with is DNA that's very low quantity and quality, and the risk for contamination is much, much higher. So when you're working with fresh tissues like blood or frozen tissues like liver or, you know, could be feathers or whatever, when you're working with fresh samples, most of the DNA is going to be intact and it's going to be very concentrated. But once you start working with things that have been long dead or collected from the field, like if you're working with scat that might have been collected from a trail out in a park, or if you're working with water samples doing environmental DNA that you're trying to screen something that's at very, very low concentration and not very good quality, that's when you really run into some problems. So if I would want to do ancient DNA projects at the museum, I would have to have a dedicated space to actually extract the DNA from those tissues and set up the initial reactions before I could bring them into the main lab. So yeah, there's a lot of sort of special circumstances when you're working with samples like ancient DNA and environmental DNA, unfortunately.
Is there, so obviously museum collections with taxidermies and study skins and things are very valuable for DNA research. Are past museum practices something that is a kind of an obstacle to overcome in terms of like how the DNA is preserved, like some of the preservatives that might have been used in things?
It could be, yeah. So there are a lot of chemicals that might interfere. So things like arsenic was a big,
I was thinking of
yeah, so arsenic was used to treat a lot of old taxidermy and study skins and collections. And arsenic could potentially interfere with genetic applications. Also, a lot of specimens were, you know, painted or dyed to enhance their colors. So that needs to be dealt with. So if you're taking, for example, we sampled our great auk taxidermy mount a few months ago, and it appeared that the feet had really been painted. And we had to get through all of that paint and those coatings to really get into the tissue to sample the actual skin. So that's another issue too, is you could have things like lacquers and dyes and paints and whatnot covering some of the samples that you might want to collect. So yeah, there could be chemical issues that would cross-react with some of the procedures we try to do in the DNA lab.
Are there any updates on the Great Auk project?
No, I'm assuming that they're shut down just like everybody else.
Yeah, that's what I would think too.
But we're waiting. We're waiting for them to get back to us. We're dying to know.
Heather, so these real-time PCR tests, I was looking at this stuff. So the ones that they're using for antibodies for COVID. So they look like a pregnancy test, so that if it's positive for it, the lines go across the little window. So my question is, I understand why they want them to be real time, but do you have any idea what is the decrease in accuracy for the real-time PCR versus having these samples sent to a lab and done? Is it more accurate to do that?
Well, the test that you're describing, I'm not familiar with that one. That one sounds like an instantaneous thing. So the test that I'm familiar with, they collect the swab. So they get your nasal secretions from the swab, and then they'll actually extract the DNA from that swab. And what they're doing is they're testing for the genetic material of the virus. Right. So what that PCR is going to do is it will basically give you a positive or negative and a relative concentration of the DNA in your sample. So you'll extract the DNA, put it into what's called a PCR, a polymerase chain reaction, that's going to target and copy any of that DNA that's present. So basically what they're looking for is the virus DNA. Actually, I think this is an RNA virus and they have to do a couple things in between, but but they're using basically that same technology. So you put it in the machine and it will give you in a couple of hours a positive or negative. And the diagnostic machines they have can actually run, I think the largest ones can run 360 some samples simultaneously. So you can run in large batches. It takes a couple of hours. And then if you have multiple machines, you can scale that up. So we've heard a lot about the scaling up of testing and being able to process more samples in the same amount of time. So the PCR tests are the ones I'm more familiar with. There might be other types of tests that they're doing now for COVID, but.
So are they just looking for just a certain strand, a segment, or a marker?
Yep.
Obviously they're not looking at the whole genome for the coronavirus. So they're looking for something particular?
Yeah, so they're looking for a marker that's specific to this particular virus.
Okay.
Because we're carrying around all kinds of viruses and all kinds of bacteria and whatnot. So they need to target specifically what they're looking for. So very, very early on in the pandemic, they sequenced the genome of the virus that causes COVID. So they had the entire DNA sequence, and what they had to do was target a specific piece of that genome that would identify the COVID infection versus anything else that we might be carrying around.
Okay.
So it is very specific, and like I said, the genome for the virus was done very, very early because people knew that that was going to have to be a tool that we used to track it. So whenever you have these emerging diseases, that's one of the first things that gets done these days is the genome is sequenced so we know what to target, not only for tests, but also for these antibody vaccines and things that they're trying to test. We need to know what is specific, what's different about COVID, the COVID virus versus other viruses that are floating around.
Okay.
That make sense?
It does.
I'm not an epidemiologist, but from my understanding, that's what's going on. So.
Cool. Well, thank you.
All right. Anything else?
Excellent job, Heather. Thank you very much.
Thank you. Thanks for coming.